We basically argued that the kidneys are filtering the blood at a very rapid rate, very aggressive rate. But curiously, just about everything that gets filtered gets taken back into the renal blood and then is redistributed around the rest of the body. So it seems to be a somewhat paradoxical process that the kidneys would utilize so much energy to filter blood and then just end up reabsorbing it right back. Now, that being the case, I'm going to talk about that while that occurs in just a moment. That being the case, all of this should look fairly familiar to you. We're looking at a renal tubule here. Notice the renal tubules right over here. It's in the proximal region of the nephron, right? Proximal region of the nephron. And make sure you get your orientation. So down here is the lumen, and then at the top there would be the blood. Now, aside from that, all of these transport mechanisms should look very similar or very familiar to you. Look, here's our little key up here. Make sure you understand that on the apical surface, the preponderance of transport, how would you describe it? What's the mechanism of transport on the apical side? Secondary active transport. Secondary active transport. So just about everything is taken in. That would include, for example, glucose, would include amino acids, would include some of these ions over here. This is our NKCC transporter, for example, which we talked about in the intestinal unit. And then, and I emphasize this because I recall several of you missed this very point on a previous test. Once you've increased the concentration of these ions and various other substances in the cytoplasm, what type of transport is appropriate for getting the substance across the basolateral surface? Yeah, be careful with your terminology here. It's going to be passive transport. Not passive diffusion, but passive transport or facilitated diffusion. But in reality, a variety of mechanisms are involved, and certainly this is the preponderant scheme in the proximal region. All of this transport is going to be dependent upon the activity of the sodium-potassium pump. In particular, the sodium-potassium pump does all those wonderful things we talked about earlier in the semester. It is electrogenic. What do we mean by the electrogenic activity of the sodium-potassium pump? Anybody recall? Electrogenic. What is it pumping? What nature of ion is it pumping? Is it pumping an anion, cation, quarks? So it's pumping all cations all day, right? Is it pumping them in equal proportion either direction? No, it's pumping 3 in one direction, 2 in the other. Remember that contributed to the membrane potential difference? So that membrane potential difference, which in part is attributable to that sodium-potassium pump, is another aspect of this reabsorption. It favors movement of, for example, ions across the membrane. The sodium-potassium pump is also important for its effects on osmosis. We talked about the fact that it's pumping a higher number of total particles outside than inside. It's going to contribute in that way. And then there's something called solvent drag. It generally comes up in discussions of the GI tract and the kidneys. And basically, it's like debris going over a dam, or water going over a dam, and little tree branches and various other substances may go across the dam. Basically, the very copious, the very large volumes of water that move across the membranes have a tendency to carry other things with them. So all of these, just reminding you, may also contribute to the transport and migration of substances across these membranes. There's a discussion here in the lower section of glucose transport. Really, this is just intended to remind you that virtually all of the glucose that is filtered is immediately taken back up. Glucose is conserved as much as possible. And there are a couple of transporters. We don't need to worry too much about them, but you should know what we mean by S-glut. What does that indicate? Secondary active transport, or sodium-dependent transport of glucose. One is a little more specific than the other. And then as we go back, so in other words, you should recognize this transporter and this transporter, know the mechanism of function for each. So again, just me, this is the table making the same point that I've made about 1,000 times already. Virtually all glucose is reabsorbed in the proximal tubule, virtually all, well, we've got to be careful with water. What we're really, let me state that what we're looking at in this table is reabsorption across the entire nephron. So most of this is done in the proximal tubule, but eventually all of the glucose, virtually all of the water, all of the sodium, virtually all the calcium. You can see the list here. The only one that really stands out is urea. And what is urea? Yeah, this is basically a waste product of protein metabolism, waste product of amination reactions, and we'll come back to that before. But in fact, at any given time, what we will argue is that about half of the nitrogenous waste is excreted, and about half, depending upon a variety of circumstances, is retained in the kidneys to maintain a very essential concentration gradient we'll talk about in just a moment. So we've seen this illustration before. We know the difference between cortical nephrons over here and juxtamedullary nephrons. And we know all about filtration. We know all about reabsorption and secretion. And in fact, you had an idealized illustration earlier, in which we just saw a little Bowman's capsule, we saw a perfectly linear nephron. And in that nephron, there was filtration, reabsorption, and secretion. So my question for you, as you look at the actual anatomy of the nephron, why is this necessary? Why is this letter N necessary? Why not just take this tubule and go right straight down to the calyx and be done with it? What is the physiological necessity of this foolishness, where it comes down and goes all the way back up and then goes down again? It just seems like unnecessary complexity. I'm sorry?
There's differences in the concentration gradient as you go down further into the cortex.
All right, so David is talking about a difference in concentration gradient. And we're going to talk about that. That's going to be our next topic. But even so, wouldn't that concentration gradient still be there if I made this direct shot down?
More surface area optimized reabsorption.
All right. If your theory is correct, why don't I just do this then?
That's probably wrong.
No, you're right. But I want you to think, because really we're just looking for the big picture here. This summer course, we don't have time to explore a lot of the details. And you'll talk about the various renal pathologies later. But what I do want you to know before you leave, before you finish the semester, is why on earth the nephron has the shape, and in more general terms, what on earth the kidneys are doing. How do they function? So let's talk a little bit about that. Now, keep in mind this general, what we're going to be talking about next is the role of the loop of Henle. So keep in mind the loop of Henle goes down, comes back up, and we want to try to explain why that's necessary in order to generate what David was talking about, what sometimes is called the corticomedullary concentration gradient, or you often read it as the corticopapillary concentration gradient. So let's talk about that. And the way I'm going to do that, in the interest of time, is I'm going to go look at a bunch of pretty pictures. So you can see, here's our idealized nephron here. We're looking at the loop of Henle. All of our discussion now is about the loop of Henle. We're addressing the question, we're asking the question, why is the loop of Henle a loop? Why isn't it just a straight tubule going all the way down, or as perhaps a suggestive convoluted tubule? All right. And I'm going to start over here. Notice where I'm starting. I'm starting on the ascending side. Remember, there's a descending side that emerges right from the renal corpuscle, and then it curves around at what's called the little loop down there, the little apex of the loop. And then what follows is the ascending side, and the ascending side will give rise to the distal tubule and eventually empty into the collecting duct. So we're over here on the ascending side. Now what I want you to notice, again, in the interest of just simplifying things dramatically, is that on the ascending side, there's massive transport of substances out of the tubule. Solutes are being actively transported in a variety of ways out of the tubule. And as we take particles, as we take substances out of the fluid in the tubule, what happens to the concentration of the filtrate? Does it go up or down?
Down.
Right, it goes down. Everybody see this? Make a note if that's not clear. As we pump the solutes out, our filtrate concentration is going to decrease. And of course, all of those particles, all of those ions and various other substances, but mostly how this works is by means of a transporter we've already talked about. This one. Remember the NKCC? It's mainly pushing chloride out, and then sodium is following along with some other things, but that's primarily what's happening. All right, so you get the general idea. Particulates, electrolytes are leaving the lumen, lowering the concentration. They move out into the interstitium here. Let's just call it the renal medullary interstitium. Medullary interstitium, where there are peritubular capillaries. Of course, down here we call them vasorecta. Now, what do you notice? In the nephron, the filtrate is moving upward. Again, we'll just use, like the universe, what direction are we moving right now in the cosmos? Anybody know? Where are we in the cosmos? But the fluid is moving up to the distal tubule, up toward the cortex. That's what we're saying. Now, which direction is the blood in the vasorecta moving? Down. So these particles are going to be leaving the nephron, entering the vasorecta, and be carried down toward the papillary region. Now, how is that going to? And of course, this is like most capillaries. The walls are fairly permeable, like most capillaries have different degrees. So as those particles are carried down, there's going to be an equalization of concentration between the blood in the vasorecta and the medullary interstitium. So the particles leaving, again, make sure you understand the big picture here because we don't have a lot of time to spend on this. Particles leaving the ascending tubule, the ascending limb, are being taken into the interstitium and then into the capillary blood where they're transported down, thereby increasing the concentration toward the papilla. All right, so let's go back to the ascending limb here. And we can look at maybe this little box right there. That'll be our exemplar. Now on this side, rather than particulates, rather than particles leaving, water. The walls of this tubule are permeable to water. All right. Now remember the concentration on the ascending side, which we'll imagine is over here, is increasing. If this wall is permeable to water, water is going to leave by virtue of the fact that the concentration is higher in the interstitium, just simple osmosis. And then here's the key thing, because this will be very confusing if you don't see this. Notice that in the peritubular capillary system here, the blood is running again the opposite direction. On the descending limb, filtrate's coming down, blood's going up toward the cortex. The water is moving into the peritubular capillary system and leaving the kidney. Because if it stayed here, we wouldn't be contributing to the concentration gradient. So it's being taken out. So the net result of those two processes, and you can sort of see what's happening here, and be aware of these numbers. We talked about these to some extent. Basically, the filtrate concentration is isoosmotic to that of the blood in the proximal tubule. And then, by virtue of this system we just talked about, particles leaving, and then those particles are going to be taken into the peritubular capillary system downward towards the papillary region. Water leaving proximally and in the descending limb, but being taken out of the renal medulla. What you're going to have then is a progressive intensification of concentration, increasing concentration. What's not shown here, and be sure you understand, is the numbers that you see in the nephron here, in the loop of Henle, are also the numbers that are present out here in the white, out here in the interstitium. So look at this. Potentially, the filtrate concentration, as it approaches the apex of the loop of Henle here, is increased fourfold. That's going to be the concentration out here too. So make sure you appreciate that. And then Remember the question I asked you earlier, does the loop of Henle concentrate the filtrate? Does the loop of Henle concentrate the urine? What happens to the concentration of filtrate as we go back up to the ascending limb into the distal tubule? Right, so it's actually more dilute. The filtrate concentration is actually more dilute as it leaves the loop of Henle. So the question then becomes, what is the function of the loop of Henle if it does not concentrate the filtrate, does not concentrate the urine? It concentrates the interstitium. But I want to make sure you understand. We'll have another picture here. I don't like that one either. Let's look at this one. And then we'll go back. So what I want to make sure you understand is that the interstitial concentrations in that medullary interstitium are roughly the same as those of the filtrate and the blood. There's some differences there to achieve these migrations that we talked about. And again, roughly the same in the tubule, in the interstitium, and in the peritubular capillary system. It's just the fluids are going in opposite directions. So that brings up the question, if the function of the loop of Henle is to generate the corticopapillary concentration gradient from 300 up there in the cortex, again, we're simplifying this, about 300 isoosmotic to blood, physiological osmolality up there in the cortex, all the way to potentially, this isn't usually realized, but all the way potentially down to 4 times physiological osmolality down in the papilla. Why? So you've created a concentration gradient. For what purpose? Anybody have any philosophical suppositions, thoughts, ruminations, cogitations?
There needs to be a two-step process of working both things at one time. Different tissue types or gradients. It's got to take out the water first, and then it can go ahead to the second step, removing the actual ions.
Oh, so you're focusing on what's, this is all happening. It's something called the counter current multiplier, and it's all happening. There's something called the first pass. We just described the first pass, and then as the fluid here becomes more concentrated, it gets even more concentrated, and it goes in a loop like this. There's a little illustration back here. But my fundamental question, let's go back to this figure. This is illustrating the counter current multiplier. Again, particles leave, enter, go down, particles leave, enter, and with each pass, it becomes more and more concentrated. But what I want you to understand, and this is not the best illustration for that, this one will work. Once the loop of Henle has created the medullary concentration gradient, what does the filtrate do? It's way back up in the, I'm exaggerating, it's potentially way back up in the cortex. And what is the fate, as you look at the anatomy of the nephron here, what is the fate of the filtrate that enters the loop, the distal tubule? What is it going to do? It's going to leave out. So the junction is not always the case, but the junction in this case is way up there near the cortex, and so the filtrate is going to pass through the medulla one more time. And what do we know about the medulla? The medulla has a very, very high concentration of solute particles. Is that clear? So the osmolality, so let's say that you're a water particle, and most of you are mostly. You're a water molecule, and you're coming down the collecting duct. What are you going to want to do? Leave. For what purpose? What's motivating you to leave? The concentration. All right, so what I want you to understand, there's a profound osmotic gradient drawing the water into the interstitium. Why would that be desirable? Why go to all the trouble? What's the purpose of the kidney?
If we didn't have that, wouldn't we lose all of our body water, our urine, and we die?
Well, you're getting a little grin here. You're supposed to be a ray of sunlight to your patients. Look, you're going to die soon, right? But let's at least understand what's happening. You're exactly right. So I just want to make sure you appreciate that. First, the whole purpose of the loop of Henle, the whole purpose of generating the medullary concentration gradient is to pull the water out of the collecting duct for the purpose of conserving water. The kidneys, one of their primary functions is to conserve water. All right, so let's go back to this. So the room here is the collecting duct. The room is the collecting duct here. We're going down, the papilla's down that way. So if indeed the water is going to want to leave the collecting duct for the medulla, what will actually determine how much water leaves by osmosis? Because that's really water can do one of two things, right? It can either, forget the urea, it can either leave and go into the medulla, or it can remain in the collecting duct and end up as what? Here. So what determines which way, what happens?
Hormones.
Because again, this is the collecting duct here. This is the wall of the collecting duct. What would determine the fate of the water? Well, particle concentration is already there. You're exactly right, and there are a lot of factors that will affect the concentration of the interstitium. But what I'm hoping you appreciate here is that the walls of the collecting duct have variable permeability to water. The walls of the collecting duct have a variable permeability to water. And they have little doors. They have little water channels. What do we call those? We talked about these earlier in the semester.
Fenster station.
Aquaport. By the way, I see some of you taking pictures. I reposted the slides yesterday afternoon and made sure that they were all there. All right, so the aquapores are these little doors that allow the water to leave the walls of the collecting duct in response to what? Why does the water leave once again? Because of the osmotic concentration gradient. It's entering up there at a concentration of 50 or 100. It's passing through. It's going to want to leave as long as two things are present. Number one, there's a profound and significant concentration gradient on the other side of the collecting duct wall. And number two, the collecting duct is permeable to the water. And what determines the permeability?
Antidiuretic?
The relative abundance of aquaporins.
Okay.
We've almost got the final step to our story here. Everybody with me so far? All right, I'll bet some of you already know. What regulates the relative abundance of the membrane proteins that we call water channels or aquaporins? How does nature add more, or open, let's just make it very simple, open more water channels in the collecting duct? Exactly. ADH. So ADH regulates the abundance. So with ADH levels, this can get 52. Make sure you understand this. As ADH levels increase, what happens to the permeability of the collecting duct? Well, increases. Finally, don't let me trick you with this, as Jeffrey was saying. What happens to the volume of urine as ADH levels go up?
Decreases.
What? Decrease. And why? What happened? Where did all the water go? Sounds like a Bob Dylan song or something. It goes into the interstitium, right? Everybody got that? So, bottom line, I want to make sure you understand, the loop of Henle does not concentrate the urine. The loop of Henle generates the concentration gradient in the corticopapillary interstitium that then enables or permits regulation of collecting duct permeability. And so really it's this final point here in the anatomy of the nephron that really is responsible for regulating the volume of water. There was another mechanism by which this occurs too. We'll talk about this in a minute.
So are the aquapores exclusively in the collecting duct?
No.
Or are they throughout the entire?
You know, they're all over the place, and I've got a figure coming up in just a moment where we'll address that. What we're going to find, remember, there's a lot of water reabsorbed up there in the proximal tubule. There's water. When you have a patient with high blood pressure, what is the first line therapy? I think it's still first therapy. Yeah. And so Bob said a diuretic, you can see a couple here, furosemide and thiazide. What are they doing? What does a loop diuretic do? Take this one, for example. Remember that channel we said in the ascending wall and the ascending limb that's responsible for pumping those particles out? What is this particular antidiuretic doing? It's blocking the transport, all right? So how is that going to change things? What does that have to do? How does that act as an antidiuretic? How does this blocking this transport increase the urine volume? Quick. Sure.
There's less particles in the interstitium, so water stays in the actual.
Yeah. Fewer particles are leaving, concentration remains fairly high here, so the volume of the water leaving is going to be altered for that reason. And up there you can see the main difference is that, for example, the furosemide group acts on the ascending limb and others act on the distal tubule. But in both cases, loop diuretics are just altering the permeability of the wall.
I'm going to piggyback on Lisa's question. Is it true that only maybe the distal tubule and the collecting duct have ADH receptors?
Yeah, that is more important. Yeah, that's a good point. And you can see that here. And that's what this diagram is actually intending to show is the sensitivity of each of these channels to a variety of drugs and substances. And so, yes, antidiuretic, ADH, and aldosterone are primarily targeting this distal region and the collecting duct more so than the prior portions of the nephron. But are there any questions about the general theory here? That's really what we're going for.
So ADH and aldosterone both kind of block the particles moving on the side?
No. So again, two different mechanisms. The loop diuretics are inhibiting transport, let's just say in the ascending limb to keep it simple. Yeah. Right, so that's one mechanism. What does ADH do? ADH is an antidiuretic hormone that works by an entirely different mechanism, as will aldosterone for a moment. What is it doing? It's altering the concentration or the abundance of transporters, aquaporins in the wall of the collecting duct.
So aldosterone blocks the
No, this is just aldosterone. All right, so what we're going to learn is that both aldosterone and ADH are antidiuretics. So what does that mean? They're increasing or decreasing the volume of the urine? Decreasing. All right, and what we're going to find is that as ADH is acting on aquaporins, aldosterone acts upon sodium transporters in the membrane. Why would a sodium transporter, what would a sodium transporter have to do with water retention? Water is going to follow sodium. So two different mechanisms there, but they both are antidiuretics. They just act by slightly different mechanisms. Right now, and we're going to come to that in a moment. I don't want to take too much time doing this because I'm not going to achieve what I wanted to today. But really what I want you to get from this is the general theory, the general nature of the general purpose and function of the nephron. Loop of Henle generates the concentration gradient. Filtrate has to pass through one last time on its way down to the papilla or urine, and that concentration gradient is essential for the regulated conservation of water, the regulated movement or transport of water through the collecting duct wall. And then there's a discussion in here about, in several of these slides, is talking about what we just discussed. If you want to a more complete appreciation. As we've indicated, one of the important functions of the kidney is to regulate the levels of nitrogenous waste, basically the products of protein digestion. And the primary product of protein digestion in humans is something called urea, NH2CONH2. And the urea levels are maintained at a fairly high concentration in the kidney, in the medulla especially, and that is necessary in order to achieve optimal concentration of the medulla. So what we're saying is the following. In order to get that medullary concentration up to 800, 1,000, 1,200, we need lots of particles in the medulla. And a proportion of those particles, by the way, also regulated by ADH, is urea. And so urea is basically there especially when large volumes of water must be conserved, bringing that concentration at the papilla or close to the papilla up to 1,200. What's really happening is, again, the urine travels through the nephron. About half of it, we talked about this before, about half of it is excreted, but about half of it remains. It's very leaky in this region, and about half of it just keeps going over and over again. So the physiological consequence of that is that it's acting to enhance or increase the interstitial medullary concentration gradient, contributing to that. So we talked about the peritubular capillaries. We talked about the countercurrent multiplier. The countercurrent multiplier, I don't know how important this is for our purposes. Countercurrent multiplier is this. Just going back in that little circle there, all right, particles. When we talk about the countercurrent exchange, we're talking primarily about the fact that all that water that would dilute, because water is leaving on the descending limb, all that water that would otherwise dilute the interstitium, what happens to it by means of the peritubular capillary system? It's taken out of the kidneys into the general circulation, and the blood that leaves. The particles that leave on the ascending limb are taken into the blood and travel downwards to enhance or increase the medullary concentration gradient. So the contribution of the vasa recta is responsible for the countercurrent exchange, which is the relationship between the nephron and the peritubular capillary system. Again, I'm more concerned that you just understand the general theory here, you understand what's going on and why it happens. Let's go to this. Just some numbers I want you to keep in mind, because you're going to need these numbers from now on. All right, remember the concentration, the osmolality in the proximal convoluted tubule is roughly the same as that of the blood, so it's going to be around 300. You should know that. And you should know that the blood entering and leaving the vasa recta is also about physiological osmolality, about 300. It's usually a little more concentrated when it leaves, but the difference is not great. What does change, as we've said several times, is the concentration of the filtrate as it leaves the nephron. Be sure you understand that it's going to be more dilute than when it entered the loop of Henle. And it can range anywhere from about 50 to 200, depending upon a variety of factors, primarily because the reabsorption in this region is highly regulated. The reabsorption in the distal tubule is highly regulated by a variety of hormones, especially with respect to some electrolytes. So hopefully then, all of this should afford some clarity. You can understand why the nephron needs to go back up. It doesn't come back down, or just doesn't go make one direct loop or one direct line to the papilla. It has to go back up, and then the collecting duct descends through one more time to facilitate reabsorption or elimination of water, depending on what is required. I might come back to that. So we've talked about the distal tubule. Just remember that by the time we get to the distal tubule, most everything has been absorbed. Most everything has been reabsorbed. But what remains to be reabsorbed in the distal tubule and the collecting duct is regulated. What do I mean by regulated? There are physiological points of control, regulatory control over, for example, magnesium, calcium, and as we've seen, water in the collecting duct. We're looking at the collecting duct here. Previously we mentioned 10 or 12 of those distal tubules will aggregate into one collecting duct. Multiple collecting ducts will tend to fuse as they get close to the papilla anatomically. And for simplicity's sake, we'll say that there are two general classes of cell that make up the wall of the collecting duct, and this will just serve as an entryway into our next discussion. There are principal cells, most important, and we'll talk about this in just a moment, most important for sodium reabsorption. These would be the targets of aldosterone, for example. And then there are also intercalated cells that are important for secretion of hydrogen ions. Because we're going to find that the kidneys play a very important role in regulating the pH of the blood. You can see some typical numbers here. We said the filtration entering the collecting duct is typically on the order of 50 to 100 milliosmoles. Those would be typical values. But it's going to vary based upon the activity, how much the person has been drinking for whatever reason, the degree of perspiration. Maximum around 20 liters a day. It's incredible to imagine that, but up to 20 liters a day. A minimum of about a half liter a day. If a person doesn't, if there isn't at least this volume of urine passing from the kidneys, then waste toxic metabolites tend to build up. So this is sort of the minimum. This is sort of the maximum that you might anticipate. Again, depending upon a variety of circumstances. Now this is an important graphic because it sort of addresses some of the questions that Luis and Bob were asking about earlier. What we're looking at here, for the most part, is how and where the water is reabsorbed along the nephron. So, very simply, most of the water, about 2/3 of the water, is taken up proximally. And there's not a great deal of regulation there. About 15% or so is subject to the activity of loop diuretics. By that I mean about 15% of the water is taken up in the loop of Henle. There's not much left, right? So here's where all the fun occurs, here's where all the action occurs in the distal tubule and the collecting duct. That leaves about 19%, because there's going to be about 1% leaving that is regulated. About 19 or 20% is regulated by those hormones we'll be talking about, ADH and aldosterone. We'll say chiefly in the collecting duct, but also to a lesser extent in the distal tubule. So is that clear? Does everybody understand the importance of this number right here? That's the target of these hormones. That percentage is the percentage of water in the nephron that is subject to endocrine regulation by ADH and aldosterone, for the most part. And this is just a summary of what we've been discussing. If you want a review of those numbers again, you should know those numbers. You should know these numbers. And then, what is misleading about these numbers? Any problem with the numbers in the collecting duct there? What's going to alter these values? The permeability of the wall, which we said is going to be influenced by ADH and aldosterone. So I've pretty much given away the conclusion of the story. You already know all about ADH. You know where it originates. You know how it's secreted. You know it's a posterior pituitary hormone. It's released in response, for example, to decreased mean arterial pressure. And also increased concentration of the blood, although while this is true, we have to be careful because the mechanism is not as straightforward as perhaps you've read in the textbooks. But those are the two primary triggers for release of ADH. So what's the theory here? Someone explain this to me. If mean arterial pressure, blood pressure is abnormally low, why would more ADH be released?
Water.
All right, so ADH is going to act to open or close the doors of the collecting duct. Shouldn't be any hesitation at this point. Open or close the doors. Open the doors. Open the doors, all right? Because when you open the doors, what's the water going to do? Like what you want to do now, you want to leave, right?
Yes, that's right.
And so the water's going to leave, and if it leaves through the doorway here, it's not going to be excreted. It's not going to be leaving as urine. And that's going to minimize further loss of blood pressure, further reduction in blood pressure, because total body water is going to be related to blood pressure. We talked about volemia earlier, hypo and hypervolemia. And the greater the total body water, what happens to blood pressure? Goes up. So if your patient has lost copious amounts of water for whatever reason, bleeding out, whatever, that could affect blood pressure. We indicated also that ADH is also going to regulate urea concentrations. Remember, the higher the concentration of urea in the medullary interstitium, the greater the concentration gradient, the greater the pull of the water through the collecting duct wall. Everybody see the general theory here? Now, I include this illustration because I want you to bring together some of the things we talked about earlier in the semester. Somebody explain what's happening in this picture. ADH is what type of hormone? What's its chemical structure? It's a peptide. So where's it going to bind? Receptor at the membrane. So no big deal here because what is aldosterone? Steroid. So where is it going to bind? Probably
Intracellular.
yeah, so note the difference here. That could help you later on. So on the left here, we have the ADH getting ready to bind its receptor. What is this thing? Yeah, the primary G protein you enjoyed so much in our previous discussions. And you can see when the ADH binds the receptor, the G protein receptor, what happens? The G protein dissociates. The alpha subunit hops over to our little sphere here, which is what? Adenylyl cyclase, causing the release of the cyclic AMP second messenger. And then things go dark, things go fuzzy, kind of like most of the time in here. And what happens? What are they showing here? So when there's binding of ADH to the receptor, what are they showing here? Yeah, more water passing through the collecting duct wall. They don't get into the details, do they? But I do want you to put it all together. This is a peptide hormone binding at a membrane receptor, triggering that our good old buddy, the G protein signaling pathway, the PKA pathway, cyclic AMP pathway, resulting in enhanced permeability of the collecting duct wall to water, transport water. Excuse me, now you have several illustrations on several graphics. We won't take time to go. I thought there were several, I think, later on, discussing how ADH works. So if you need a graphic discussion of that, there you go. A couple of pathologies you should be familiar with, SIADH and diabetes insipidus. I think I've asked you this question before. What do all forms of diabetes have in common? What do all forms of diabetes have in common? No. You wouldn't argue that for mellitus, for example. Abnormal urination. And that's what we're talking. We're talking about essential diabetes insipidus here. It has nothing to do with sugar, at least not directly. This simply results from abnormally low ADH levels, and that could be for any number of reasons. Oftentimes the cause is not known, but these patients will exhibit polyuria. What is that?
Lots of being.
Yeah, urinating too much. Polydipsia? Yeah, you're right. That's a proper clinical way. They're thirsty most of the time. Nocturia? Yeah, you get my age, you know. Yeah, urinations at night. But generally what there is a loss of hypothalamic regulation. In SIADH, I don't know if you can see this little illustration up here, one of the common causes of inappropriate release of ADH is small cell lung cancer. This seems to be associated with ectopic release of ADH. What do we mean by ectopic? Yeah, in a location that is abnormal. And you can see in the illustration, you have it from the brain. You also have ADH coming from the brain. You have ADH coming from the lungs here. In fact, this is a very common indicator of small cell lung cancer. And as a result, the individual may suffer, for example, abnormal water retention, hyponatremia, and so All right, let's look at the second of these two hormones. This one's a little more complicated, but in general it does the same thing. So we're going to be talking about the renin-angiotensin-aldosterone system. Recall we talked about the juxtaglomerular apparatus, so we're looking at the afferent and the efferent arterioles here. Here's the distal tubule coming right back up by that Bowman's capsule we talked about before. You can see the macula densa cells here, and these are sensitive to both the volume, the pressure in the distal tubule, and the osmolality. Although, whatever you've read in the textbooks, that mechanism is extremely complex, and we won't get into it here. We're happy to talk it, and you're not responsible for it on the test. It will take us a couple hours to work through that. But the general idea here is that these cells that you see right here, these granular cells or juxtaglomerular cells, will release the hormone renin, and renin is a renal hormone, in response to sympathetic stimulation or in response to reduced blood pressure, arterial hypertension. Now, before we take our break, notice the little figure down here in the lower right. What does renin do? Renin is a hormone that's responsible for converting that horrible thing that Jeffrey was talking about earlier, angiotensinogen. By the way, what do you know about any word that ends in OGEN? It's a precursor. It's inactive. It's a preliminary inactive form into angiotensin 1. All right, so renin converts the precursor into a semi-active form. And then in the lungs and the kidneys, the lungs of all places, the lungs and the kidneys, angiotensin converting enzyme converts angiotensin 1 into angiotensin 2, which is physiologically extremely active. So why don't we stop there, and we'll talk about what angiotensin 2 does when we come back from the break.